Section 18.1, How Animals Reproduce. By the end of this section, you will be able to describe advantages and disadvantages of asexual and sexual reproduction, discuss asexual reproduction methods, discuss sexual reproduction methods, and discuss internal and external methods of fertilization. Some animals produce offspring through asexual reproduction, while other animals produce offspring through sexual reproduction. Both methods have advantages and disadvantages. Asexual reproduction produces offspring that are genetically identical to the parent because the offspring are all clones of the original parent. A single individual can produce offspring asexually, and large numbers of offspring can be produced quickly. These are two advantages that asexually reproducing organisms have over sexually reproducing organisms. In a stable or predictable environment, asexual reproduction is an effective means of reproduction because all of the offspring will be adapted to that environment. In an unstable or unpredictable environment, species that reproduce asexually may be at a disadvantage because all the offspring are genetically identical and may not be adapted to different conditions. During sexual reproduction, the genetic material of two individuals is combined to produce genetically diverse offspring that differ from their parents. The genetic diversity of sexually produced offspring is thought to give sexually reproducing individuals greater fitness because more of their offspring may survive and reproduce in an unpredictable or changing environment. Species that reproduce sexually and have separate sexes must maintain two different types of individuals, males and females. Only half the population, females, can produce the offspring, so fewer offspring will be produced when compared to asexual reproduction. This is a disadvantage of sexual reproduction compared to asexual reproduction. Asexual reproduction. Asexual reproduction occurs in prokaryotic microorganisms, bacteria and archaea, and in many eukaryotic, single-celled and multicelled organisms. There are several ways that animals reproduce asexually, the details of which vary among individual species. Fission. Fission, also called binary fission, occurs in some invertebrate multicelled organisms. It is in some ways analogous to the process of binary fission of single-celled prokaryotic organisms. The term fission is applied to instances in which an organism appears to split itself into two parts and, if necessary, regenerate the missing parts of each new organism. For example, species of turbellarian flatworms, commonly called the planarians, such as Dugacea dorotocephala, are able to separate their bodies into head and tail regions and then regenerate the missing half in each of the two new organisms. Sea anemones, Cnidaria, such as species of the genus Anthopleura, will divide along the oral-aboral axis, and sea cucumbers, Echinodermata, of the genus Holothuria, will divide into two halves across the oral-aboral axis and regenerate the other half in each of the resulting individuals. Figure 18.12. The Anthopleura artemisia sea anemone can reproduce through fission. Budding. Budding is a form of asexual reproduction that results from the outgrowth of a part of the body leading to a separation of the bud from the original organism and the formation of two individuals, one smaller than the other. Budding occurs commonly in some invertebrate animals, such as hydras and corals. In hydras, a bud forms that develops into an adult and breaks away from the main body. Figure 18.3. A. Hydra reproduce asexually through budding. A bud forms on the tubular body of an adult hydra, develops a mouth and tentacles, and then detaches from its parent. The new hydra is fully developed and will find its own location for attachment. B. Some coral, such as the Lophelia pertusa, shown here, can reproduce through budding. Fragmentation. Fragmentation is the breaking of an individual into parts followed by regeneration. If the animal is capable of fragmentation and the parts are big enough, a separate individual will regrow from each part. Fragmentation may occur through accidental damage, damage from predators, or as a natural form of reproduction. Reproduction through fragmentation is observed in sponges, some cnidarians, turbolarians, echinoderms, and annelids. In some sea stars, a new individual can be regenerated from a broken arm and a piece of the central disc. This sea star is in the process of growing a complete sea star from an arm that has been cut off. Fisheries workers have been known to try to kill the sea stars eating their clam or oyster beds by cutting them in half and throwing them back into the ocean. Unfortunately for the workers, the two parts can each regenerate a new half, resulting in twice as many sea stars to prey upon the oysters and clams. Figure 18.4. A. Lynchia multiflora is a species of sea star that can reproduce asexually via fragmentation. In this process, B. An arm that has been shed grows into a new sea star. Parthenogenesis. Parthenogenesis is a form of asexual reproduction in which an egg develops into an individual without being fertilized. The resulting offspring can be either haploid or diploid, depending on the process in the species. Parthenogenesis occurs in invertebrates such as water fleas, rotifers, aphids, stick insects, and ants, wasps, and bees. Ants, bees, and wasps use parthenogenesis to produce haploid males, or drones. The diploid females, workers and queens, are the result of a fertilized egg. Some vertebrate animals, such as certain reptiles, amphibians, and fish, also reproduce through parthenogenesis. Parthenogenesis has been observed in species in which the sexes were separated in terrestrial or marine zoos. Two female Komodo dragons, a hammerhead shark and a blacktop shark, have produced parthenogenic young when the females have been isolated from males. It is possible that the asexual reproduction observed occurred in response to unusual circumstances and would normally not occur. Sexual reproduction. Sexual reproduction is the combination of reproductive cells from two individuals to form genetically unique offspring. The nature of the individuals that produce the two kinds of gametes can vary, having, for example, separate sexes or multiple sexes in each individual. Sex determination, the mechanism that determines which sex an individual develops into, can also vary. Hermaphroditism. Hermaphroditism occurs in animals in which one individual has both male and female reproductive systems. Invertebrates such as earthworms, slugs, tapeworms, and snails are often hermaphroditic. Hermaphrodites may self-fertilize, but typically they will mate with another of their species, fertilizing each other and both producing offspring. Many species have specific mechanisms in place to prevent self-fertilization because it is an extreme form of inbreeding and usually produces less fit offspring. Hermaphrodite is not an accepted term for humans and does not describe differences in their sexual development. Intersex people are those whose traits or reproductive anatomy develop differently from the typical ways humans develop and can include hormonal, chromosomal, or anatomical differences. Figure 18.5. A. Many snails are hermaphrodites. When two individuals, shown in B, mate, they can produce up to 100 eggs each. Sex determination. Pioneering scientist Nettie Stevens was the first to observe chromosomal differences between the different sexes of organisms. Using a microscope to observe mealworm cells, she noted that one chromosome was notably different between females and males, and she concluded that those chromosomes were the most likely determinants of the worm's sex. She later studied other insects, and her discoveries were confirmed by other scientists. Stevens was initially denied credit due to her gender, but later publications acknowledged her critical role. Mammalian sex is determined genetically by the combination of X and Y chromosomes. Individuals homozygous for X, as in XX, are female, and heterozygous individuals, XY, are male. In mammals, the presence of a Y chromosome causes the development of male characteristics, and its absence results in female characteristics. The XY system is also found in some insects and plants. Bird sex determination is dependent on the combination of Z and W chromosomes. Homozygous for Z, ZZ, results in a male, and heterozygous, ZW, results in a female. Notice that this system is the opposite of the mammalian system, because in birds, the female is the sex with different sex chromosomes. The W appears to be essential in determining the sex of the individual, similar to the Y chromosome in mammals. Some fish, crustaceans, insects, such as butterflies and moths, and reptiles use the ZW system. More complicated chromosomal sex determining systems also exist. For example, some swordtail fish have 3 sex chromosomes in a population. The sex of some other species is not determined by chromosomes, but by some aspect of the environment. Sex determination in alligators, some turtles, and tuataras, for example, is dependent on the temperature during the middle third of egg development. This is referred to as environmental sex determination, or more specifically, as temperature-dependent sex determination. In many turtles, cooler temperatures during egg incubation produce males, and warm temperatures produce females, while in many other species of turtles, the reverse is true. In some crocodiles and some turtles, moderate temperatures produce males, and both warm and cool temperatures produce females. Individuals of some species change their entire set of reproductive organs during their lives, switching from one to the other. If the individual is born with an ovarian system of organs first, it is termed protogeny, or first female. If it is born with a testicular system of organs first, it is termed protandry, or first male. Oysters are born with male morphology, grow in size, and change body parts and lay eggs. The wrasses, a family of reef fishes, are all sequential hermaphrodites. Some of these species live in closely coordinated schools with a dominant male and a large number of smaller females. If the male dies, a female increases in size, changes sex, and becomes the new dominant male. Fertilization. The fusion of a sperm and an egg is a process called fertilization. This can occur either inside, internal fertilization, or outside, external fertilization, the body of the female. Humans provide an example of the former, whereas frog reproduction is an example of the latter. External fertilization. External fertilization usually occurs in aquatic environments where both eggs and sperm are released into the water. After the sperm reaches the egg, fertilization takes place. Most external fertilization happens during the process of spawning, where one or several females release their eggs and the males release sperm in the same area at the same time. The spawning may be triggered by environmental signals, such as water temperature or the length of daylight. Nearly all fish spawn, as do crustaceans, such as crabs and shrimp, mollusks, such as oysters, squid, and echinoderms, such as sea urchins and sea cucumbers. Frogs, corals, squid, and octopuses also spawn. Figure 18.6. During sexual reproduction in toads, the male grasps the female from behind and externally fertilizes the eggs as they are deposited. Internal fertilization. Internal fertilization occurs most often in terrestrial animals, although some aquatic animals also use this method. Internal fertilization may occur by the male directly depositing sperm in the female during mating. It may also occur by the male depositing sperm in the environment, usually in a protective structure, which a female picks up to deposit the sperm in the reproductive tract. There are 3 ways that offspring are produced following internal fertilization. In oviparity, fertilized eggs are laid outside the parent's body and develop there, receiving nourishment from the yolk that is a part of the egg. This occurs in some bony fish, some reptiles, a few cartilaginous fish, some amphibians, a few mammals, and all birds. Most non-avian reptiles and insects produce leathery eggs, while birds and some turtles produce eggs with high concentrations of calcium carbonate in the shell, making them hard. Chicken eggs are an example of a hard shell. The eggs of the egg-laying mammals, such as the platypus and echidna, are leathery. In ovoviparity, fertilized eggs are retained in the female, and the embryo obtains its nourishment from the egg's yolk. The eggs are retained in the female's body until they hatch inside the body, or the female lays the eggs right before they hatch. This process helps protect the eggs until hatching. This occurs in some bony fish, like the platyfish Xiphophorus maculatus, some sharks, lizards, some snakes, the garter snake Thamnophis sartalis, some vipers, and some invertebrate animals, like the Madagascar hissing cockroach Gromphodorina portentosa. In viviparity, the young are born alive. They obtain their nourishment from the female and are born in varying states of maturity. This occurs in most mammals, some cartilaginous fish, and a few reptiles. Figure 18.7. A. In oviparity, young develop in eggs outside the female body, as with these Harmonia axidridis beetles hatching. Some aquatic animals, like this bee, pregnant Xiphophorus maculatus, are ovoviparous, with the egg developing inside the female and nutrition supplied primarily from the yolk. In mammals, nutrition is supported by the placenta, as was the case with this newborn squirrel, shown in C.